Good evening, everyone. Welcome to the Dry Eye Club Seeing Outcomes webinar 2024. Painful dry eye. What do we know? My name is Dr. Maria Cabrera Aguas. I am a research associate at the Cornell Research Group at the University of Sydney SafeSide Institute. I will be your moderator today. First of all, before the event starts, we want to recognize and pay respect to the elders and communities past, present, and emerging of the lands that the University of Sydney's campuses stand on. For thousands of years, they have shared and exchanged knowledges across innumerable generations for the benefit of all. This is the agenda for today's webinar. First, we're gonna have some housekeeping rules. Then later, we're gonna have some polls. We have 4 amazing talks today, followed by a Q&A session. First, I would like to point some housekeeping rules. If you have any question, please post it on the Q&A box down below. And we will have the Q&A session, as I said, after the presentations. During this webinar, we will launch 2 polls. The first poll will be launched before the first speaker and the second before the third speaker. At the end of the webinar, we will ask you to kindly complete a feedback survey for this webinar. And within the next week, we will send a link of the webinar recording to all of the registrants. So let's continue. Great, thank you for participating. So let's introduce our first speaker. And our first speaker is Professor Stephanie Watson. She's the Chief Investigator of the SafeSide Dry Eye Registry, Head of the Cornell Research Group, SafeSide Institute, the University of Sydney, Head of the Cornell Unit at the Sydney Eye Hospital, Chair of the Australian Vision Research, and Vice Chair of the Asia Pacific Ophthalmic Trauma Society, and Vice Chair of Bransco New South Wales Branch. She will present on the SafeSide Dry Eye Registry. Thank you. Welcome, Stephanie.
Thank you very much for the kind words of introduction. I'm just going to start by sharing my screen. And so in this talk, I'm going to give a little overview of the SafeSide Dry Eye Registry and how we're using it to find the evidence to improve outcomes for patients with dry eye disease. So first of all, look, many of you probably know a bit about dry eye, given that you're on the webinar, but just to remind you that it's really a very common problem with the prevalence across the globe ranging from about 5 to 50% in some countries. And it's actually on the increase with more modernization, more use of computer screens, air conditioning, and also people living longer because it gets worse with age. Now, there's a range of treatments available for dry eye disease, but despite this, there's many patients that still continue to suffer. And we still don't know, why that is for all, and we don't know which treatments are best. And so there's an unmet need to collect data on dry eye disease from everyday practice. And this is so we can learn more about which treatments work best for who, and also to help clinicians benchmark their own practice. And so having information on the outcomes of treatments and comparing them from one clinician to the next enables improvements in practice. So, and the reason why it can be complex to treat dry eye is this diagram here shows you the different range of conditions that actually can cause dry eye disease. And this is just a summary, so there's more than this. And there's also different types of dry eye disease, so much so that treatment now needs to be tailored to individual patients' needs. So in medicine, when we treat a condition, the best way to select a treatment is to use evidence. And that's evidence on how safe and effective the treatment is. And one of the ways of getting evidence is to perform studies, and there's a range of different studies. And registry studies fall into this, on the pyramid of evidence here, cohort studies. And so they're halfway up the pyramid or a little bit closer to the top, and they can be used to help us figure out what treatments work and are safe. and they're observational in nature. So what that means is that patients are using a treatment and they're just observed. There's no intervention. Patients aren't asked to use the treatments in a randomized trial in a very strict way. In an observational study or registry, they use the treatments like they would in everyday care. But because of this, the results can be a little bit confounded because there might be many factors that influence outcomes. Therefore, we need large amounts of data to overcome these confounding factors. So an example of a cohort study that we did, or a couple of cohort studies, was looking at women on aromatase inhibitors for the treatment of breast cancer. And we found that they actually suffered with dry eye, but many didn't realize it was associated with treatment and didn't seek help. But what we're here to talk about today, or I'm here to talk about in my talk, is clinical registries and the Safe Sight Dry Registry. And this is because it's been recognized in ophthalmology that registries can actually provide a lot of this cohort data. It can help us by collecting observations on patients and finding out more about treatments and how they work. And to this end, we have the Safe Sight Registries. It's a group of registries that were first started by Professor Mark Gillies for retinal diseases. And I now lead the Fight Corneal Blindness Project, which focuses on corneal diseases. Now, why we need registries is, and I've alluded to this already, but in randomized controlled trials, what happens is you take patients. You only take certain patients. You have inclusion and exclusion criteria, and then you apply a treatment in a very strict way, and then you make very specific observations over time. And so it's a bit like Mr. Perfect down here, but the real world is a little bit messier than that because patients use treatments in different ways, and patients have comorbidities as well. Also, with clinical trials, they generally run for short periods of time, 3-6 months, whereas with a registry, you can collect data over the long term. So I established the Dry Eye Registry in 2020. It was launched in 2021. COVID was still happening, so not an ideal time, but it's now grown to over 1,000 eyes. And this is just an example of some of the information that we can learn from the registry. This is a study where we use the registry data from over those 4 years. And we had patient demographics, medical history, clinical tests. We also collected from the patients information about their dry eyes. So how this works is with the registry. There's questionnaires that can be completed, and Dr. Kandel will talk more about this later. And this information gets fed into the registry. So the clinician not only sees the state of the eyes in a physical sense, but importantly hears from the patient about how the dry eye is affecting them. And what we found out is that in everyday practice, evaporative dry eye is the most common. So around 84% of people have dry eye due to the tear quality not being good rather than not having enough tears. And this is important to know because managing tear quality is different to managing tear quantity. We also found that many patients had a range of symptoms, but most of the symptoms were sort of moderate to severe. So patients can be suffering quite badly with their dry eye symptoms. And many also reported some degree of anxiety or depression. So this data was collected over a number of centres, and it was good to see from this study that the registry really was an easy-to-use tool, and it was able to facilitate the collection of large amounts of data so we can learn about dry eye. And the registry is continuing to collect data. You'll hear more about some of the studies later on in the webinar. But the aim is really to have enough data to be able to compare different treatment outcomes for different treatments that are available, and also to understand more about the natural history of dry eye. Does it get better? Does it get worse? Who does it get better in? Who does it get worse in? And in what circumstances that occurs? Now, we do still need randomized trials. And these studies, though, as a complement, or the registry complements the randomized trials, because the randomized trials will evaluate new treatments, but they are resource intensive and expensive. An example of a recent randomized trial was our publication in the Ocular Surface, where we looked at a new treatment for meibomian gland disease and found that this was quite successful in improving some of the features. And so this trial provided data which will now help the treatment then be further developed to reach the clinic. So going back to our pyramid, we have our cohort studies, our registries, which I hope I've described as useful for gaining evidence on dry eye and its treatment. Randomized trials also useful, but many people also like to use things like artificial intelligence. And it's important to know where this fits into the evidence pyramid. So we did a study looking at ChatGPT and using it as a tool for conducting a literature review for dry eye disease. And so we asked ChatGPT to find the 10 most cited articles for dry eye. And what we found that ChatGPT brought up articles, 60% of those didn't even exist, and only 30% matched with a single prompt. And there were a number of errors in those articles in that some of them didn't have the right identifiers. And this could be because ChatGPT doesn't have access to some medical databases. And we even went to ask ChatGPT whether they were sure they were correct, and it replied, "I strive to provide accurate information to the best of my abilities based on the information available to me. However, if there is any doubt about the correctness of the articles I have provided, I encourage you to verify them yourself through a reliable source." And so remember, using these sort of artificial intelligence types of chat box and sometimes information on the internet may not give you the quality of the evidence that you need for your dry eye decisions. Okay, so just to sum up, the SafeSight Dry Eye Registry can inform on findings from the real world, what really happens, and we can find out about dry eye treatment safety and effectiveness. We can also understand the patient perspective, and we'll hear more about that later. And it can add to data that's available from randomized trials. I'd just like to close by thanking my research team shown here, others that have supported my research, the SafeSight Registries team, and there's a QR code there you can scan to find out more. Thank you very much.
Thank you, Stephanie. That was a great introduction to the SafeSight Dry Eye Registry. So now let's move on to the next talk. So the next presenter is Dr. Ngozi Chidi-Egboka. She's a postdoctoral research associate and clinical coordinator for the Cornell Research Group at SafeSight Institute. She will present on dry eye disease treatments and therapies in everyday practice. Welcome, Ngozi.
Thank you, Maria, for the introduction. So I'm going to start by sharing my screen.
Yes.
Okay, sorry about that. So as highlighted by Professor Watson, the burden of dry eye disease is enormous, and it's mostly multifactorial, often caused by environmental, ocular conditions, or systemic conditions. And patients affected by dry eye disease often adopt different types of treatment, like artificial tears, cold compress, use of wraparound sunglasses, different forms of lid hygiene, and also environmental modifications such as avoiding air conditioning, using humidifier, and also trying to stay hydrated, and often some behavioral strategies like blinking more frequently. With the Fight Corneal Blindness Project, we track the real-world evidence on these patient treatment options to generate outcomes and understand how to improve the ocular health. And so we can track the diseases, the dry disease diagnosis of patients that come to see their doctors on everyday practice, and their dry disease can be diagnosed based on the clinician judgment as evaporative, aqueous, or the mix of both. We also keep track of all the medical treatments that are in the registry from which the clinician can select which one their patients are using, prior treatment or current treatment. We also keep track of the therapeutic procedures such as mechanical, surgical, or physical treatment. For example, the use of punctal plugs. This is a typical example of real-world data on medical treatment used by patients recorded in the Safe Side Dry Eye Registry. We have a range of dry eye disease treatment, but for the purpose of this webinar, I'll be focusing my talk on topical cyclosporine. So topical cyclosporine are medications approved for use in the treatment of moderate to severe dry eye disease, where dry eye is not controlled adequately by the use of artificial eye drops. And so in Australia, we have a range of products, a range of cyclosporine products, including Icovis, which is listed in the Pharmaceutical Benefit Scheme in Australia in October 2021. We also have Sequa, which was PBS listed from June 2023. We also have Restasis, which was listed, which is not PBS listed, but it is available in Australia through the Special Access Scheme or the authorized prescriber. We also have compounded cyclosporine. This is often prescribed based on clinician decision, especially when patients are showing intolerance to their commercially available products or they're having adverse reactions to the products. It is important to note that cyclosporine is a prescription-only medicine in Australia. Only ophthalmologists or therapeutically qualified optometrists can prescribe this medication. In order to qualify for the Pharmaceutical Benefit Scheme subsidy, patient needs to meet some criteria. And for initial prescription, there has to be some dry symptoms and signs for eligible patients, and they also have to show that their dry is not being controlled by tissue tears. And then for patients to be eligible for continuing treatment under the PBS subsidy, there has to be the prior treatment with under subsidy, and also there has to be evidence that the dry symptoms and signs have improved by at least 30%. It's important to follow up on patients every 6 months, and the maximum amount of repeats that could be available under the PBS scheme is 5 repeats. Cyclosporine can also be available as a private script in Australia, whereby patients do not need to meet the PBS guidelines for the prescription, and they are unable to access the PBS subsidy, and usually dosed in accordance with the product information. So talking about the product information, cyclosporine is approved only for use in adults over 18 years old, where dry eye disease is not controlled. I must emphasize again, with artificial tears. It's mostly available as single ampoules and preservative-free, whereby patients are advised to discard each ampoule after each use. And then for the dosing, depends on the product. For the Icovis, it's usually once daily as final medication at bedtime. And then the Sequa and the Restasis are used twice daily. The compounded products used as prescribed by your treating doctor. When do we expect initial response from treatment? Usually within 4 to 12 weeks, based on evidence in the literature, in the publications and everyday practice. We also keep track of improvements in the real world using the Safe Side Dry Eye Registry. And so this is how the tracking goes. We keep track of the procedures, we keep track of the treatments and the therapeutic interventions, we keep track of adverse events. And this is helpful for clinicians to be able to assess their outcome against other doctors, and also to be able to audit their clinical practice in compliance with continuing professional education activities. A visual summary of the procedures and the treatment and the outcome over visit date of each patient for each eye can be instantly available in the Safe Side Dry Registry. And it's a very important tool for patients to be involved in their treatment journey as well. And we also keep track of adverse events. On the topical cyclosporine side, evidence from most publications in the literature shows that the reactions are mostly mild and are mostly related to topical reaction to instillation. So mainly pain, irritation, stinging, burning that patients do feel when the drops are instilled. They often resolve requiring no treatment. and the report of discontinuation of the drugs due to this event is quite minimal. I must emphasize again that cyclosporine is not currently approved for use in individuals under 18 years old. However, in children over 3 years old, under Sydney Children's Hospital Network, it can be used for cases of serious or severe infectious conjunctivitis. It's also important to note that the use of cyclosporine with contact lens in place in the eye is not recommended. So remove your contact lenses before using the drops and wait at least up to 15 minutes before you reinsert your contact lenses. Avoid contact with the eye with the tip of the ampoule to avoid the risk of injury or eye infection. It is not also recommended for use with pregnancy. So in conclusion, the Safe Side Dry Registry is a very good tool that facilitates the collection of large amounts of clinical data on everyday practice or treatment given to patients with dry eye disease. Treatment is usually safe with cyclosporine, and in general, usually there is no adverse event reported. So I must end by acknowledging the expert panel that have contributed to developing the practical guidance for use of topical cyclosporine. I would also like to thank the Safe Side Registry team and the steering committee. And for clinicians wanting to be users of the Safe Side Dry Registry or Safe Side Registry in general, the QR code here, you could also send us an email to the contact. Thank you, everyone.
Thank you, Ngozi, for your talk. Let's continue with our next speaker. So our next speaker is Dr. Himal Kandel. He's a Kornhauser Research Fellow at SafeSide Institute, University of Sydney. He will present on the usefulness of patient-reported outcomes in dry eye. Welcome, Himal.
Thank you, Maria. Hello, everyone. Really happy to talk about patient-reported outcomes, an area that I believe is very important. First of all, what are patient-reported outcomes? The FDA defines patient-reported outcomes as any report of patient's health status that comes directly from the patient without interpretation by the clinician or the researcher. Patient-reported outcomes include quality of life domains, such as symptoms, physical, emotional, and social functions. Patient-reported outcomes are collected or evaluated using the PROMs or PRO measures, most often using the validated questionnaires. The examples of PROMs or the validated questionnaires I'm using today is from our SafeSide Dry Eye Registry. We have 3 validated questionnaires in the registry. The first one is the Ocular Surface Disease Index, OSDI, and there are 12 items or 12 questions in 3 domains of quality of life: activity limitation, symptoms, and environmental triggers. The questions are formatted very similar to the polls we did earlier. The next questionnaire we have in the registry is the OCI, or the Ocular Comfort Index. The OSDI is the most frequently used questionnaire globally in dry eye management. And OCI, recent research has shown that OCI actually performs better than OSDI. So we in the registry, it doesn't take a long time to complete the question. So we have been collecting data using OSDI as well as OCI. OCI also has 12 items on 6 symptoms. Frequency and intensity attributes of each symptoms are evaluated using the OCI. The third questionnaire we have in the registry is a patient health questionnaire. This is actually, if you look at the content of this questionnaire, the content is not specific to dry eye, but it is for evaluating the mental health status it could be used in any conditions, but in the registry, of course, we use it to evaluate the mental health status in dry eye disease. We also, in the registry, we also collect patient-reported outcome data on screen time and medication use, which using these 2 questions here. This is not part of the validated questionnaire, but also equally important is this can impact on the dry eye disease outcomes. In the registry allows the real-time measurement. What it means is, as soon as the data is fed into the registry, the scoring is done automatically, and the clinician can immediately see the quality of life scores in different domains, and they can immediately compare with the scores in the previous visits. So it's a very useful clinical tool to evaluate what is happening to patients in terms of patient-reported outcomes. Now coming to the main part of the topic today, the importance or the usefulness of patient-reported outcomes. The pros enhance communication between patients and clinicians. They are often the conversation starters, and they give more voice to the patients. Often patients visit hospitals or clinicians when they have a lot of issues, but the prompts or the questionnaires allow us to understand more subtle issues and more comprehensively in various domains of quality of life. It is also an opportunity to involve patients in their own care, and patients provide valuable information about their condition. They know the best, and as they have been living with it. The regulatory bodies such as the FDA recommend that prompts or the validated questionnaire should be part of the comprehensive outcome assessment, complementing the objective outcomes clinical tests that we do in the clinics. Other important, it's really important to evaluate prompts to respect patient values and interest. For example, there are various kinds of refractive surgeries available, which may all offer good visual outcomes, but it is really important to consider the patient perspective as well, like probably the surgery which gives better outcomes in terms of symptoms, that less symptoms will be better for patients. So it is pros are important to respect patient values and interest. The final very important use of pro data, especially in our website dry registry, is for research. It has been very useful resource to inform us to understand more about the dry eye disease impact, as well as to find the ways we can improve the outcomes. For example, in this recent study, we found that the signs and symptoms in dry eye disease, were not correlated or weakly correlated, which means that we need to evaluate both signs as well as symptoms, and we can't predict signs from symptoms or vice versa. So this is a really important finding. Similarly, we could do subgroup analysis. Here are some examples again. Like we found that patients who had mixed dry eye disease, mix of evaporative as well as aqueous deficiency dry eye disease, they had worse quality of life scores compared to the dry eye disease patients who had only evaporative type. Similarly, patients with corneal neuropathic pain had worse quality of life scores compared to other patients. So in conclusion, patient-reported outcomes are the outcomes that are most valued by the patients, and they are essential for increasing the value of our healthcare. Thank you very much.
Thank you, Himal, for introducing us to the patient-reported outcomes. Thank you. So now we're gonna move on to our next speaker, is Professor. I'm gonna share my. Can you stop sharing your screen? Did you?
I think I did.
Yeah. Okay. Sorry. Yeah. There you go. So our next speaker is Professor Laura Downie. She's a professor and Dame Kate Campbell Fellow for Research Excellence in the Department of Optometry and Vision Sciences at the University of Melbourne. She will present on current challenges in relation to accurate diagnosis and phenotyping of dry eye in the clinic and next generation technologies to advance care for dry eye disease. Welcome, Laura, to our webinar today.
Thank you. Okay, is that coming through okay?
Perfect. Yeah.
Okay, so thank you to the Save Sight Sydney team for the opportunity to contribute today. I'd also like to begin by acknowledging the traditional owners of the land from which I join you today, which is the Wurundjeri people of the Kulin Nation, and pay my respects to elders past and present. My disclosures are as shown, and these are relevant to industry funding for my laboratory at the University of Melbourne. So I'm an optometrist and a clinician researcher, and I lead the Anterior Eye Clinical Trials and Research Translation Unit. So we aim to advance knowledge about the biology of the ocular surface. We also aim to improve how we can detect and monitor disease with different markers of eye health and evaluate new treatments to improve eye care. And this is really across three main areas. The first is how we can use information from the eye as a window to health. And so the focus here is analysing tears and also imaging immune cells in the eye. And it's these two topics that I'm going to focus on today. We also undertake clinical trials, like Stephanie mentioned, to really improve treatment for eye disease. And this involves testing new drugs and devices, including for dry eye. And importantly, we also aim to help how we translate evidence into practice. And this relates to developing resources for clinicians so we can provide the best care to our patients. So clearly, dry eye involves changes to the tear film, which sits at the surface of the eye and is a key protective fluid layer for the ocular surface. So whilst tears are mostly water, we know that healthy tears are actually very complex with up to 2,000 unique components. And this includes a range of different proteins and even salt components, which we recognise as electrolytes. So in a healthy tear film, there is this exquisite balance of these different components, but in dry eye disease, we observe relative imbalances in this tear composition. And so one of the common definitions of dry eye is that it is a multifactorial disease of the ocular surface, characterized by a loss of homeostasis of the tear film and accompanied by ocular symptoms, with various clinical signs also being important to understand the etiology. So there's three key points from this slide though, and the first is about this concept of dry eye being multifactorial. So it recognises that dry eye is complex and can't yet be fully characterised by a single sign or symptom in the clinic. Also central to dry eye is this loss of tear film homeostasis. And so again, this reflects that there can be many possible changes and imbalances in the tear film, and this will disrupt the integrity of that ocular barrier. And finally, as we've just heard from Himal, ocular symptoms and patient experience is also a key component of the definition. And so we know many patients will experience a spectrum of different symptoms. This might include discomfort, burning, or even pain in some instances. So now we've considered this overall definition, as we also heard earlier, there are different types of dry eye. And at the kind of most fundamental level, there are two main forms that are recognised: aqueous deficiency, where we don't produce enough tears, so there's a lack of tear volume, and evaporative dry eye, where the tears evaporate off the surface too quickly. And the most common reason for this is lipid abnormalities. So we have a lipid layer on the very outermost surface of the tear fluid composition, and this is secreted by specialized glands in our eyelids known as meibomian glands. And so the reason it's so important to know which subtype or which element of a subtype a patient has is that it's really critical for informing which type of treatment is likely best suited to that patient. And so for aqueous deficiency, in many cases, patients will require anti-inflammatory drugs or measures that preserve the tear on the eye. Whereas for evaporative dry eye, here our treatment is largely focused on improving the function of those lipid-secreting eyelid glands and may involve antibiotics. So stratifying these patients is very important. So how do then we as clinicians aim to diagnose dry eye and its subtypes so we can then prescribe our patients the most appropriate therapies? So this slide shows the various techniques that are available to us, and we know that these on their own have various strengths and limitations. But broadly, they aim to characterize different aspects of tear film health. So we might look at the tear film breakup time as shown here to look at tear stability. We can quantify how much tear a patient produces using measures like the Schirmer test. We can analyse the electrolyte composition using measures like tear osmolarity, and even we can evaluate the downstream effects of tear dysfunction by looking at different types of ocular surface staining on the far left. But in many cases, these techniques really only show us one aspect about the health of the tear film and the ocular surface. So despite this broad range of clinical tests, there are still challenges to the optimal dry eye diagnosis in the clinic. And we've been working with optometrists in Australia and the United States to really understand what these challenges are. So the top 3 challenges that have been identified by clinicians are as follows. First, relating to diagnosis of dry eye, it's estimated that many patients don't receive a comprehensive diagnosis because there isn't really a single gold standard test. We also know, as we just saw for that excellent data from Himal, that there's often a lack of correlation between the measurements we take in the clinic, the science, and what patients feel. So there is a mismatch between dry eye symptoms and signs. The second challenge relates to accurate subtyping. So it's currently quite challenging to reliably separate aqueous deficient from evaporative dry eye, and therefore to be able to target the main etiological factors that might be causing a patient's presentation. And then the final challenge is in relation to treatment. So how do we optimally match what we see in the clinic to a given therapy? And ideally, what we'd be able to do is match the mechanism of action, or how the therapy is working, to ideally align with the cause of a patient's dry eye. So with these challenges, though, we have opportunities to develop new technologies that address these problems and can improve patient care. And so an optimal scenario could be that we would attain an accurate diagnosis, including the subtyping information, with our patient at the point of care, to then ensure we have the information to prescribe the right treatment to the appropriate patient, and then optimize their health and quality of life outcomes. So with this in mind, our research team are working to develop some new indicators that can be used to also correlate with a patient's symptom level in dry eye disease. So we're seeking to improve how dry eye is diagnosed and subtype, so that a clinician can readily identify what's causing a patient's tear deficiency and then prescribe a targeted treatment that appropriately addresses the lipid problem or the inflammation or both. And so I'm going to briefly share 2 examples of technologies that we're currently working on. So the first technology is called ADMIRE, which stands for Acoustically Driven Microfluidic Extensional Rheometry. And it works by measuring the viscoelastic properties, or the stretchiness behavior, of a tear droplet. So we collect a microliter tear sample from a patient's eye and then quantify the stretchiness behavior of that sample over a matter of milliseconds to achieve a comprehensive characterization of that patient's tear film health. What we've found is that the viscoelastic properties, or the stretchiness of a tear, are reduced in someone with dry eye disease. So tears from people with dry eye have a lower viscoelasticity than healthy tears. We also find the extent to which that tear viscoelasticity is reduced relates to the patient's severity of dry eye. So more severe dry eye will have a lower tear viscoelasticity. To date, we've done 2 clinical studies and compared how the ADMIRE technology compares relative to a series of clinical tests that would be considered a gold standard diagnostic protocol. And we're really excited that the technology shows very high accuracy to identify dry eye, can also differentiate between a lipid and aqueous deficient form, and really importantly, the markers that we measure in the tears actually correlate with dry eye symptoms. So this looks as a very promising way that we can get an objective readout of a patient-reported outcome. So we're now working to develop the technology to be used at the point of care so clinicians can rapidly and accurately diagnose and subtype dry eye, and then give that targeted drug therapy and monitor how the patient responds to that treatment. The second example is a new imaging method that we've developed to measure immune responses in the cornea in patients. And so the cornea is this, we know, is the dome-shaped outer surface of the eye. It's transparent for clear vision and also one of the most sensitive parts of the body. And so you can see in the lower image here, we have a region of the cornea known as the whorl from a human. And we can see there are many of these linear features, which are sensory nerves. The cornea also provides one of the first lines of defense for the eye against infection. And so it's home to many immune cells. And the reason we're so interested in the cornea is it's the only place in the body where we can directly see these sensory nerves, as well as these different types of immune cells in a living human. So to image the human cornea in this way, we use an instrument known as an in vivo confocal microscope. For the patient, they have a topical anesthetic eye drop placed, and then there's a gentle non-contact device that touches the front of the eye, and we can capture really high resolution scans. So here we have an image of the corneal epithelium, and so we can see the nerves in yellow, as well as the immune cells shown in the green circles. And until recently, this type of imaging was very much limited to what we can see here, so-called static images. So single images which don't tell us about the behavior of the cells. And so we've been working to overcome this challenge by combining this type of imaging with the principles of animation. And we've developed a technique that we call functional in vivo confocal microscopy, or FUN-IVCM. So to look at the immune cell dynamics, we take images of the same region of cornea every 5 minutes in patients for about 30 minutes. We then align them to the nerves, which don't move, and then overlay them to form a video. So we think of this a bit like a flip book animation, as shown here. So when the individual images of the cornea are viewed in very quick succession, the parts of the image that have changed location will be seen to move. So by doing this, we're able to see different types of immune cells moving in the corneas of our clinical patients. So here's another example. So this is what we call a time-lapse video. It's taken over about 20 minutes from a human patient. And you'll see that there are kind of two main types of cell behaviors. In the middle of the video on the left, there is this small motile cell that we now recognize as an adaptive immune cell, a T cell. And then there's also these much larger cells called dendritic cells that don't move their cell bodies, but instead use their dendrites to survey the local environment. So what's exciting about this method is that we've been able to show that the dynamics of these different cells change differentially with different types of inflammation. And also, really importantly, when we treat patients with anti-inflammatory drugs, we can actually quantify a normalization of how the cells move that aligns with what we see clinically. So this imaging technology gives us a means to measure the effect of drugs on immune responses in the eyes of our patients. So we're now interested in how we can use this to identify patients who have more severe inflammation and potentially different types of inflammation based on imaging that shows us how their immune cells move. We're interested in whether we can use this technology to inform different types of treatments for dry eye, and really think there's scope to better understand the immune response by tracking patients over time. So in conclusion, we know dry eye is common, and there is a need to improve how we diagnose it in the clinic to inform targeted treatments. There's a lot of research into technologies that can identify dry eye, subtype the condition, and then allow us to provide more targeted treatment to provide better care for our patients. So I'd just like to acknowledge many people who contribute to the work in our lab in the Department of Optometry at the University of Melbourne, including Dr. Holly Chinnery, who co-leads the Frontier Research Centre, now from Perth, with myself, as well as our lab members, our collaborators, and the funding for the research. Thank you very much.
Thank you, Laura. Amazing talk. Amazing all the projects you are doing.
Thank you.
We can keep talking all night about it. All right, so our final speaker is Rachel Love, and she works in medical philanthropy at the University of Sydney. So Rachel, welcome.
Thank you very much, Maria. I will just share my screen. There we go. Maria, is that in slideshow view?
Yeah, yeah.
Lovely.
Perfect.
Okay, great. Look, thank you very much for the introduction, Maria. And I'm very happy to be invited to join you here this evening and share some insights about the role of philanthropy in research. I'd like to start by sharing the current funding landscape. In Australia, iResearch is primarily funded through a combination of public and private sources. Public funding comes from government agencies such as the National Health and Medical Research Council, who provide grants and fellowships to support researchers. The National Health and Medical Research Council only managed to fund approximately 11 to 13% of all grant applications they receive. This forces many talented researchers to leave the sector, taking with them invaluable knowledge and skills built up over many years of work. I'll now share some of the challenges that our researchers face when it comes to funding their work. We know it has been and continues to be difficult to secure dedicated, prioritised funding for research into anterior eye conditions. Yet such conditions, one of which is dry eye disease, continues to impact increasing numbers of individuals globally. About 1 in 4 adults in Australia, as mentioned by the previous speakers. Sorry, that's come out of slideshow. There we go. There are limited resources available for research, and funding organisations must allocate their researchers across a wide range of medical conditions and scientific disciplines. Therefore, dry eye research is competing with other important areas of medical research for funding. The cost of research can also be a significant challenge. Cutting-edge research, especially clinical trials or genetic studies, can be expensive, and a lack of awareness can translate into less public support and consequently less funding. Addressing these challenges requires a collaborative effort from government, private sector, and non-profit organisations to raise awareness, allocate resources effectively, and promote research and development in the field of dry eye research. So how can philanthropy help? Public advocacy and education are crucial in garnering support for funding research, and we are delighted that so many of you have joined us here this evening. Non-profit foundations and philanthropic individuals can play a significant role in funding dry eye research here in Australia. Support from donors can play a hugely important role, and it is vital to bridging the current funding gap. Secure, longer-term sources of funding help to ensure that we attract and retain the best minds, that researchers can shape their research agenda based on the greatest need and the most immediate impact rather than on the funding available. Mid to long-term research projects can be designed and undertaken which yield more detailed and meaningful data. For example, the Safe Sight Dry Eye Registry, which some of the speakers mentioned earlier this evening, was established to provide real-world data on dry eye natural history and to inform on the outcomes of treatments for dry eye disease in the longer term. The work done by the registry also requires funding to train students in research to enable them to provide future solutions to reduce the growing burden from diseases such as dry eye. Secure funding means that research can be accelerated as consistent progress is made so the research isn't start-stop. Philanthropic funding allows research to prioritise dry eye research. With a growing population and an increasing number of people with dry eye conditions, it is vital that our researchers are able to continue this important work. Thank you for your time this evening. If anyone would like to learn more about how you can support this research, please get in touch. My details are on the screen. And you can also scan the QR code if you would like to make a donation to this research. Thank you very much.
Thank you. Thank you, Rachel, for your kind information. We have a couple of questions, so I invite all of our speakers to turn on your cameras. And we can. So let's start. Well, one of the question was maybe Stephanie or Ngozi can answer, is as a patient, how can we know if your doctor is participating in the registry?
Oh, the best way is to ask them, and they'll be able to tell you if they're participating. So, and it can get the conversation going. They may not know about it, and we're happy to provide more information. But yeah, just have a chat with them.
Okay. This is a question for Himal. So my doctor does not ask me to fill out such questionnaires. What should I do?
Yeah, I think symptoms, I mean, especially symptom management is important, especially in dry eye. And if your doctor doesn't ask to fill those questionnaires, then probably the outcome assessment is incomplete. So one way to address that it would be like ask them to tell them about the registry where we are tracking symptoms in a very systematic way. One of the reasons they're not tracking symptoms could be it is sometimes cumbersome in the clinics, if they don't implement the symptoms questionnaires well, it can lead to clinician burden or respondent burden, as well as it is not practical to like calculate scores, the symptoms scores in the clinics. We have addressed all those limitations and challenges in the registry. The automatic scoring is there. They can immediately get the scores and that kind of thing. So I think it's like Stephanie said earlier, it would be good to talk to the clinicians about what registry can do and how their symptoms can be tracked. And for the clinicians, the registry is available free of charge, so clinicians don't have to pay anything to be part of the registry. And it's a great tool to track and monitor symptoms as well as signs.
Great, perfect. Thank you, Himal, for that. And Laura, when would we be expecting to have this amazing tool to diagnose dry eye disease? Well, the type of dry eye disease. When do you think will be available?
So speaking of funding, that's probably our most critical factor at the moment. So we're still developing the technology, and really there's a major step to take something from a research laboratory prototype all the way through to clinical approval and marketing. So there's many steps to go through, and so we've made a lot of progress on the technology and validating what it can do clinically. But it's likely several years before this would be a device in the clinic. So that development of new technology and drugs often takes decades. So we'll keep working on it, but it is a long road to eventually get it in the clinic is our goal.
Oh, okay. Thank you. Thank you for that. Yeah, because sometimes as a patient, you don't know how long it takes to develop something. You may think, in a couple of years, we'll have this amazing tool. Finally, we have a question for Ngozi. How long does it take for dry eye condition to be completely treated with, for example, cyclosporine, or is it expected to be an ongoing therapy?
That's a very good question, Maria. So treatment with cyclosporine is meant to be reassessed by the clinician at least every 6 months to determine the ongoing treatment, the outcome, to be able to decide how the treatment regime is going to continue. So it's not something you could say that works similar for everyone. But from clinical trials as well perspective, some studies have done for 6 months, some have done for a year, and there has not been any established difference on the range of treatment and how the duration of treatment and what impact it has on the outcome. So my overall suggestion would be keep seeing your doctor, that will keep reassessing your eye condition to know the treatment routine to stick to.
Great, thank you. Thank you, Ngozi. Thank you for clarifying that. So, well, now we are getting to, well, we are past like 2 minutes from 6 o'clock now, but thank you everyone for attending to this webinar. Amazing talks. We got an update of the dry eye registry. We got some new research in dry eye from Laura from Melbourne. So now I really enjoyed that. I hope you also enjoy it. And again, I would like to thank our speakers, and then, well, join us for next year webinar. Have a great evening, everyone. Thank you.